Good afternoon. Welcome to the 3 Minute Thesis Finals. My name is Joe Canella. I'm with the Graduate College. I'm here to welcome you to our competition tonight. If you're unfamiliar with what 3 Minute Thesis is, and you just kind of walked in off the street, I'm going to go over a quick rundown of what 3 Minute Thesis is. It's an academic competition where doctoral candidates share their research in 3 minutes or less. And they're challenged in a unique way. They're challenged to present their research in non-academic terms so that the public can really understand what it is they do. And the reason for this is so that we can prepare our students to go out into the world and tell their stories. Universities around the world participate, and we've had over 40 entries this year into our program, or until this competition. Our preliminary competition was held back on March 3rd. We had 30 candidates, and tonight we're going to have the top 10 from that group participate. So we're really looking at the cream of the crop, some of the best communicators we have. Our students tonight will be judged in two primary areas. The first is comprehension and content. You can see the summary of these bullet points on the screen. The second is engagement and communication. The students will be scored on a scale of 10 on each of these areas and also ranked by our judges tonight. Once those scores are tallied at the end, we will look at them and select our top 3 students. Our first place winner, as well as our 2 honorable mentions, will go on to our showcase round at the reception tonight, where you can actually vote on a People's Choice winner. So I'd invite you to come to the reception tonight and have your voice heard. This is a democracy. So with that, I'm going to get started, and I'm going to introduce Andrew to introduce our next speaker.
So my name is Andrew Metzger. I'm a master's student, so I wasn't able to participate this year in 3 Minute Thesis, but I was excited to at least get involved in organization a little bit. So our first speaker is going to be Kathryn Cather. Kathryn Cather is a 4th-year graduate student in the Behavioral Neuroscience program. Originally from Plainfield, Illinois, she earned a Bachelor of Science from Bradley University in Peoria, Illinois, before moving to the Hawkeye State, and her research focuses on the neurobiology of addiction, with a particular emphasis on the contribution of the prefrontal cortex to reinstatement behaviors in a rodent model of addiction. Outside the lab, she enjoys baking, going on adventures with her husband, and learning new things from her 6-month-old budding scientist, Rosalind. Her talk is The Role of the Insular Cortex in Cocaine Relapse.
Thanks. One of the things I find most interesting about being an addiction researcher is that every time I give a talk, like this, someone usually comes up to me afterwards and says that their mother, brother, uncle has suffered from addiction. Now, while I admit at first this was a bit jarring, it always serves to remind me that the work we do in our lab isn't just about rats giving themselves cocaine. It's about uncovering what brain regions make it nearly impossible for an addict to avoid taking a drug. To do this, I use a rodent model of addiction, where rats are placed into a chamber like this and allowed to press a lever for cocaine. Along with each cocaine infusion, they are also presented with a short light and tone cue. As you can imagine, over time, rats begin to associate this light and tone cue with the drug, much in the same way that an addict may associate drug paraphernalia with their drug of choice. Now, once rats are stably responding and reliably receiving cocaine infusions, they are moved on to the next stage. At this point, cocaine is withheld from the animal, similar to when cocaine is withheld from an addict when they enter a treatment facility. Now, at this point, when rats are placed into the chamber, pressing the previously cocaine-paired lever will have no consequence. No light and tone, no cocaine. It's a complete dud. So, over the course of a few days, rats learn this change and press the lever at much lower rates. And it is at this point that we can then study relapse. Many addicts report that when they return home from treatment, simply seeing the cues they once associated with their drug of choice makes it nearly impossible to avoid taking the drug. To mimic this situation, Following treatment, our rats are once again presented with the previously cocaine-paired light and tone cue. And even though they are not receiving cocaine, just seeing these cues is enough to increase their lever pressing or cause relapse. So, I decided to investigate a region known as the insular cortex, which has previously been shown to be involved in nicotine addiction, as well as a variety of drug-associated cues, to determine if this region is involved in cued relapse. To do this, I inactivated the structure by injecting a pharmacological agent into the structure immediately prior to one of these cued sessions. And what I found is that inactivating the insular cortex led to a decrease in lever pressing during one of these cued sessions. In other words, rats without a functioning insular cortex were completely able to avoid relapsing. In contrast, inactivating the insular cortex did not affect other types of relapse, such as cocaine prime. This information is extremely valuable, as it is the first to demonstrate that the insular cortex may be one of those regions that makes it nearly impossible to avoid taking a drug. And we hope that this information will lead to treatments that are more specific and more successful.
As scientific tools have advanced, microbiologists have demanded better ways to understand DNA. Differential geometers, such as myself, are among those who have attempted to answer this call, and our goal was to build a mathematical model that could be used to represent DNA. As a pure mathematician, my focus has been on the mathematical aspects of such a model, rather than the direct applications. Now, despite what your high school textbooks may have illustrated, DNA actually spends the majority of its time in a circular form, that is, having no loose ends. As you can see here, a mathematical knot is a closed curve in three-dimensional space. This makes knots a very natural starting point in our attempt to model DNA. However, it's important here to realize that DNA is not really a perfectly smooth piece of string or wire, and therefore our use of knots as a model falls somewhat short, as they do not account for DNA's non-uniformity. Therefore, we propose the idea of a weight function. A weight function gives us the ability to match DNA's varying thickness by specifying values along the length of a knot. Intuitively, we like to think of these weighted knots as being a piece of wire along which some sort of gel representing the weight has been spread. Here I'm using red to represent the heavier areas and blue to represent the lighter ones. Of course, we would like to be able to compare weighted knots, but here a picture can only tell us so much. In a way, it's like shopping for a car. Photographs tell you part of what you would like to know, but ultimately you want some quantitative information like fuel efficiency or horsepower. This is where mathematicians have designed a variety of knot-specific statistics called knot energies. The Mobius energy is one of the most important knot energies. Defined in the early 1990s, the Mobius energy is roughly correlated to the complexity of the knot and is defined through a rather complicated formula. Part of our work was generalizing this formula so that it also allowed weighted knots. And you can see here, for example, this weighted trefoil has a Mobius energy of about 560. Returning now, though, to the car analogy, it's clear that if you change the car's tires, it could affect its fuel efficiency, but changing something like its paint color would not. We want to ask similar questions about the Mobius energy, specifically, how will adjustments in our weighted knot affect the Mobius energy? And that's been the focus of much of our research. One of the simplest variations we've considered is called the equitotal variation. Here you can see we've kept the core curves the same, we've kept the total weight the same, but we've allowed the weight's distribution to change. We've found that when you pile all the weight in one spot, it actually causes the energy to fall somewhat, whereas piling it in multiple places causes the opposite effect. The weight rises, or the energy rises. Whereas allowing it to spread out rather evenly, unsurprisingly results in a rather average-looking energy value. Of course, there's many other variations we've considered that we don't have time to discuss today, but ultimately the hope is that someday this research could build a model that would contribute to our understanding of DNA.
Our next speaker is Shi Yi Wang. Shi is a PhD student in molecular physiology and biophysics. Her hometown is Shanghai, China, located along the coast of the East China Sea. She received her Bachelor of Science degree from the University of College London in the United Kingdom, and her PhD study focuses on the functions of calcium ion channels in the brain. Besides her studies, she is interested in politics, economics, and history. Hiking and having intellectual conversations are her most joyful activities. The title of her talk is Preventing Suicide by Understanding Brain Disorders.
Each year, about 1 million people in the world die by suicide. That means one about every 40 seconds Too often, suicide is not considered as a medical issue, but it should be. Most people who committed suicides are diagnosed with brain disorders like depression, bipolar disorder, schizophrenia. While mortality for heart disease has decreased by 63% since 1995, suicide mortality rate has remained the same for decades. It is time to prevent suicide by understanding brain disorders. The brain is an organ of serial complexity. Inside the brain, billions of neurons form highly integrated networks. They function through electrical signals. These signals control nearly everything we do, from thinking to moving muscles, from sensing to responding. Even now, as you listen to my talk, your neurons are doing what they are supposed to do, keeping you in your seat instead of walking out. In recent years, neuroscientists identified some candidate genes that may cause brain disorders. CACNA1C is a gene for a type of voltage-dependent calcium channels. Genetic disruption in this gene was found in people with brain disorders. In the brain, this particular calcium channel admits calcium ions into the neuron in order to send electrical signals. Here are the questions that I really care about. How do problems in this calcium channel lead to brain disorders? How many working calcium channels does the brain need for normal function? In other words, is there a specific number of functional calcium channels required for brain to perform normally? During my PhD study, I used genetically modified mouse brain as a model of brain disorders. I found a protein called densin that acts to maintain the number of functional calcium channels in the brain. When densin is removed, the number of functional calcium channels reduced by 50%. The mice are anxious, and they have schizophrenia-like behaviors. Does that mean in order to treat brain disorders, we really need to restore these calcium channels in the brain? This is the puzzle I want to continue to figure out. In the end, I wish one day my brain research will make a life-saving change for people suffered from brain disorders. Thank you.
Our next speaker is Crystal Wotipka. Crystal is in her 5th and final year as a PhD student in communication studies. She took her bachelor's degree from the University of Texas at Austin and her master's degree at Texas State University. Her research focuses on the processes and outcomes of conducting interpersonal relationships in computer-mediated channels, and she is especially interested in online dating, as well as how people communicate difficult messages to others. When she is not busy with academia, Crystal enjoys lifting weights, thrift store shopping, and attempting to perfect complicated and interesting new recipes. The title of her talk is Embracing the Screen of Mediated Environments, an exploration of the buffer effect's role in communication surrounding transgressions.
Bad news. You've done something wrong. You've really messed up this time, and worse, you have to tell your friend about it. Your hand drifts to the cell phone in your pocket, and you wonder, should I text them now? You know you're going to see them in an hour, but you worry about how they're going to react to what you have to say. So what do? My dissertation explores this problem by examining one feature of mediated environments called the buffer effect. The buffer effect refers to a literal and figurative shield that mediated environments provide. And it's theorized to be most influential when people are communicating something difficult, like a transgression. Different channels have different levels of the buffer effect. So for example, text messaging has a high buffer, instant messaging has a medium buffer, and face-to-face has a low buffer. And so in this study, I hypothesized that senders of transgressive messages would really prefer to communicate in those environments with a high buffer, but receivers would prefer environments with a low buffer because they thought, well, maybe my friend took the easy way out. I had people come into the lab to complete an experiment, and halfway through the interaction, I had one partner say that they couldn't complete the study. They were doing the experiment for extra credit, and so saying that they couldn't complete the study rendered their partner ineligible for that extra credit. So what happened? Did the sender, the one that was theorized to prefer a high buffer, actually experience more positive outcomes in that high buffer text messaging condition? Results suggested that yes, they did. In fact, the senders were more likely to enjoy the interaction and also indicated that they were more likely to use the same channel to deliver a similar message in similar situations. However, for the receiver, the buffer effect did not make much of a difference. That is, the receiver did not experience higher levels of satisfaction, enjoyment, trust in their friend, or lower levels of hurt, regardless of the channel. What did make a difference for the receivers was how their friend portrayed the message. For example, if senders took a high level of responsibility, then the receivers were more likely to have these positive outcomes, and they actually did experience more satisfaction, more enjoyment with the interaction, more trust in their friend after the interaction was over, and less hurt. So, in sum, when sending a transgressive message to a friend, you might be more likely to be more influenced by the channel. But when receiving that message, it's actually your partner's qualities of that message, such as the responsibility level that they take, that make a difference. Thank you.
Our next speaker is Allison Song. Allison is a fourth-year graduate research assistant in the Wynn Institute for Vision Research at the University of Iowa. She is studying how to use stem cells to develop cures for age-related macular degeneration in Dr. Bud Tucker's lab. Originally from Saskatoon, Saskatchewan, Canada, Allison graduated from Abilene Christian University in Texas with a major in biochemistry and a minor in Spanish. Although she spends most of her time focusing on her stem cell research, she also enjoys classical singing, hiking, and expanding her culinary skills. The title of her talk is The Vision to Cure Blindness.
Helen Keller once said, "The only thing worse than being blind is having sight and no vision." My vision is that one day we will ultimately cure age-related macular degeneration. The thing that happens in AMD is that the macula starts to die. And the macula is a part of the eye that's the central part of the retina. And so if you will, please join me and close your left eye, make a tight fist, and bring your fist up to your right eye so you see just a little halo of light. I bet you can imagine me very frustrated trying to go about your daily activities with this kind of vision. But what you're actually seeing is the vision of someone with late-stage AMD. In the United States alone, there are 2 million people with late-stage AMD, and an additional 7 million with early-stage AMD. Unfortunately, once the macula has died, it's too late for patients to regain their lost vision, because right now there is no treatment available to replace the cells that died in their retina. But what cells die in AMD? Well, recent evidence from the University of Iowa shows that the blood vessels behind the retina are actually the first that die off in AMD. The cell types that make up those blood vessels are called choroidal endothelial cells, and they're critical to the retina's health because they nourish the retina. So I'm sure you can imagine that once those cells die in AMD progression, the other two cell types of the outer retina, called the photoreceptors, which you guys probably know as the rods and cones, and the retinal pigment epithelium, which maintain those rods and cones, those die off as well. So what can we as researchers do to help AMD patients regain their lost vision? Well, that's where my research comes into play. When I joined the Tucker Stem Cell Lab here at U Iowa, researchers in the field were already figuring out how to use patient stem cells to make healthy photoreceptors and retinal pigment epithelium in hopes to one day rebuild retinas for patients. But no one had been able to make stem cell-derived choroidal endothelial cells, those cells that make up those blood vessels that die off first in AMD. So to solve this problem, I took patients' skin cells and did something called cellular reprogramming, in which I made a patient's skin cells into their own stem cells. I then sifted through over 200 proteins to feed these stem cells to see if I could get them to become these choroidal endothelial cells to make that vasculature behind the retina. What I found was that there are two specific proteins that drove the stem cells to develop into the choroidal endothelial cell vasculature, the same cells you see on the screen. This method is the first of its kind in the eye research field. And now, by using this method, we can finally make all three cell types that die in AMD. And now we can start focusing on rebuilding patient retinas and restoring vision to those who thought they would never see again. So, like Helen Keller, I too believe in a vision, and sight is my vision. Thank you.
Our next speaker is Kather Ahmed. Kather is a fifth-year graduate student in the College of Pharmacy. She holds a bachelor's degree in pharmacy from the University of Baghdad College of Pharmacy in Iraq. She came to the U.S. in 2011 for a master's degree in pharmacy and received it in May 2013. Kather continued for PhD in the same department and is graduating this summer. Her research focuses on drug delivery for cancer immunotherapy and cancer vaccines. Outside the lab, she is a mother of 3 children and enjoys spending time with them. The title of her talk is Engineered Cancer Vaccine.
Cancer, the second cause of death in the United States and globally. The main problem with cancer is that at some point, it will stop responding to treatment. And that is when the disease starts spreading, eventually leading to death. Today, I will talk to you about a cancer vaccine formulation that I have been working on in my research with the aim of helping cancer survivors. Let me first introduce the concept of cancer vaccines. Cancer vaccines differ from conventional vaccines, which are given to protect healthy individuals from getting the disease. Cancer vaccines are given to cancer patients to protect cancer survivors from disease spreading. Now, vaccines use the immune system to fight. But in order for the immune system to fight, it first needs to recognize that there is a danger. And the problem with cancer is that the immune system does not see tumor cells as anything dangerous, as you see in number 1 here, and it does not respond to them. Cancer vaccines change the status quo. Cancer vaccines tag the tumor cells, put red flags on the tumor cells, telling the immune system, hey, this is dangerous. You need to do something about it. How do they do that? They do it by incorporating molecules called immune adjuvants, as you see in number 2 here. And these immune adjuvants, they are simply danger signals. These are red flags that the immune system will never miss. It will always respond to them because it perceives them as an infectious entity. Now to the exciting part, which is the cancer vaccine that I have been working on. So in vaccines, we use a harmless component of the disease-causing entity to trigger the immune response. In my vaccine, I use a dying tumor cell, and I'm working with prostate cancer. So I would take a prostate cancer tumor cell, I irradiate these cells, so they're dying, they cannot form a tumor anymore, and then I surface engineer the cell, meaning I modify the surface of the cell. And I do that using particles, and that's what you see in number 3 here. So that big thing, this is the tumor cell, and these little balls where the arrows are pointing, these are the particles. And I make these in the lab, and they are filled with the immune adjuvant, the danger signal that I talked about. So I attach the particle on the cell, and then I take this whole construct and inject it into the animal. And voila! Vaccinated animals were more protected against prostate cancer, and they survived longer. In other words, we have made a successful cancer vaccine formulation. So the take-home value of my research is, besides setting up a new platform for cell surface engineering, which is a big achievement on its own, the most important outcome is that we have made a promising cancer vaccine formulation that we hope it's going to help us in the fight against cancer and help protect cancer survivors. Thank you.
Our next speaker is Amber Bates. Amber is a second-year PhD student in oral science at the University of Iowa, where she previously completed a BS in human physiology and a BS in psychology. Originally from the small town of Guthrie Center, Iowa, Amber now works in microbiology immunology lab in the College of Dentistry under the supportive mentorship of Dr. Kim Brogdon. Her research focuses on matrix metalloproteinases, enzymes that break down tissue, and their role in inflammatory diseases like periodontal disease and oral cancer. The title of her talk is A Method to Study Inflammation in the Mouth.
Hi, today I'd like to talk to you about periodontal disease, a chronic inflammatory disease that affects approximately 46% of U.S. adults. In our mouth, foreign microbes form a layer of bacterial cells, or plaque, and our immune system's response to this is an inflammatory response. When this gets prolonged, it causes a state of imbalance, and certain enzymes called MMPs are increased. They act like biological scissors that degrade the tissue surrounding our teeth. If left untreated, this leads to periodontal disease, which can result in bone loss and even tooth loss. Now for my research, I want to learn more about this inflammatory response by studying these enzymes, MMPs. To do this, I have used a multi-cell system with an added agonist Hag-B, a protein from a periodontal-associated bacteria. Now this multi-cell system consists of T cells and dendritic cells, two cell types known for their immune cell functions, and then keratinocytes, which are cells from the lining of our mouth. Now this system is unique because rather than the standard method of growing one cell type by itself, we're growing 3 cell types together, and they're allowed to communicate with one another because their signals can go through a porous membrane here and here. And so that way we can study the effect the cells are having on each other. So for my experiment, I grew each cell type by itself, in pairs, and then all 3 together. And then I added my Hag-B, and after 64 hours, I determined the concentration of 9 different MMPs produced by the cells. Here's one of those MMPs that had significant differences between the groups. So what we see here is that dendritic cells are the main responder of this, the main producer of this MMP, while the other 2 cell types are not. Now, when paired with either of the other 2 cell types, there's still a very high response coming from the dendritic cells, whereas when paired with all 3 cell types, we see a different response. So what this tells us is that the cells are responding differently when they're grown by themselves compared to when they're all grown together. So as we continue this research, we want to use a multi-cell system because it's going to better represent the host environment where many different cell types are present. So my lab hopes to use this multi-cell system to do 3 different things. We're hopefully going to use it to identify other MMP inhibiting products, and then learn more about the inflammatory system of the mouth. And thirdly, in time, we hope that it will help us identify potential treatments to inflammatory diseases like periodontal disease. Thank you.
Our next speaker is Sebastian DePasquale. Sebastian is a member of the University of Iowa's Electric and Magnetic Field Instrument Suite and Instrument Science team, using NASA's Van Allen probes mission to study the coupled nature of the plasma extension of Earth's atmosphere with the sun. As a fourth-year graduate student in the Department of Physics and Astronomy, he has worked on several team teaching projects to redesign undergraduate experiments in the classroom. Sebastian owes his commitment to research and teaching from Grinnell College, where he graduated in physics with honors and the department's H. George Apostle Prize. The title of his talk is Voyage of the Van Allen Probes on the Shore of Space.
Here is my view of Iowa City and the arcing big and blue sky that we all know. I am fascinated that we are sometimes treated to such rich colors as orange streaks and purple hues. To me, these are the colors of plasma, and I imagine a site of inspiration to the study of limitless space. What we call the fourth state of matter, a collection of ions and electrons known as plasma, is quite temperamental here on Earth. This picture was taken with the sun setting from the rooftop of Van Allen Hall, which I had pictured here at the top right corner. This building was named after the native Iowan and space pioneer James A. Van Allen, who pioneered the United States' first scientific payload on the Explorer 1 rocket, and also contributed to the first space discovery, that Earth, like the sun, is surrounded by plasma. Though another Iowan, Captain James T. Kirk, followed in spirit with the Starship Enterprise, and together these 2 intrepid explorers launched a generation of dreams, their spacecraft are not the first things of this Earth to have escaped into space. At great heights above the surface, our atmosphere absorbs energetic radiation from the sun, freeing electrons and ions from gas molecules that with enough energy can diffuse outward into space by overcoming Earth's gravity. These charged particles are affected by the electromagnetic force and drift along Earth's magnetic field from pole to pole. They also mutually affect each other with this force, interacting with the surrounding environment as a region of dense plasma known as the plasma sphere. Much like a cloud is indicative of the weather on Earth, the plasma sphere responds to the geomagnetic equivalent of a storm in space. Plasma escaping from the sun's own atmosphere constantly impacts and exchanges energy with Earth's enveloping magnetic field, blown back by the solar wind. With the guidance of my Trekkie advisor, Professor Craig Kletzing, I have focused my thesis work on the role of the plasmasphere in space weather as it expands and recedes against Earth's atmosphere like a wave crashing on the shore. Professor Kletzing continues Iowa's rich tradition in space physics by leading an instrument team for the Van Allen probe's mission, which returns to the site of Explorer 1's discovery, that Earth and space is filled with plasma. My research contributes to our understanding of how the plasmasphere dynamically evolves under the influence of geomagnetic storms, and is the view afforded by the unprecedented measurements taken by the Van Allen probes. That is why I am so fascinated by the molecules we can see on Earth, here as clouds drifting in Iowa's beautiful sky, for they are a reminder of our connection, Iowa's connection to historic voyages in space. Thank you.
Our next speaker is Kathryn Peter. Kathryn is a third-year PhD student in environmental engineering. Originally from Cleveland, Ohio, she did a BS in chemical engineering at Washington University in St. Louis. Her research focuses on development of nanomaterial-enabled technologies for drinking water treatment, and she is more broadly interested in water treatment, emerging contaminants, and advanced technologies for drinking water and stormwater treatment. Outside of the lab, she is a member of Iowa City's Spanish Book Club, a volunteer at Habitat for Humanity, and enjoys quilting and hiking. The title of her talk is Nanomaterial-Enabled Drinking Water Treatment Technologies.
High-quality drinking water is essential for your everyday peace of mind. Unfortunately, our drinking water supplies are contaminated with a range of pollutants, from organics like pharmaceuticals and pesticides to heavy metals like the lead contamination in Flint and the ongoing occurrence of arsenic in Iowa's groundwater. Thus, especially in your kitchen, at your tap, there's a growing need for safe and affordable drinking water treatment technologies that can remove a wide range of contaminants. My research focuses on the development of nanomaterial-enabled technologies to fill that gap. Nanomaterials are things like nanotubes and nanoparticles that are less than 100 nanometers in size, so about 100 times smaller than a single human hair. Because nanomaterials are so small and are also highly reactive, they could be used in a very tiny reactor that could treat a lot of water. Design of that reactor is the main challenge in applying nanomaterials practically for drinking water treatment. It would be unsafe to give you a laboratory-style suspension of nanomaterials for your kitchen, and you would probably complain. It also wouldn't make sense to pack nanomaterials into a Brita-style filter because it would require a ton of pressure to push the water through, making it economically not feasible. My research uses a technique called electrospinning to design a material that contains nanomaterials and can be used practically. In electrospinning, a polymer solution is loaded in a syringe connected to a metal needle, and a high voltage difference pulls polymer nanofibers from the needle onto a grounded collector, depositing a non-woven nanofiber filter. That non-woven nanofiber filter is nanoscale, so it can be used in a very small reactor. And because it's non-woven, you can push water through very quickly without using a ton of pressure. We also can include things like nanoparticles in that polymer precursor, making a hybrid nanocomposite, where we have nanoparticles embedded in each of our nanofibers. The nanoparticles in that hybrid composite are the active agent. They're doing the work of the water treatment. That's especially exciting because it means not only are we containing the nanomaterials within our nanofiber matrix, but we're also using their excellent properties and capabilities for water treatment. Ultimately, we can combine these nanomaterials in a composite nanomaterials in a modular super unit that would have different layers intended for treatment of different types of pollutants with different types of nanomaterials in each layer. This will ultimately make nanomaterials feasible for use in your kitchen for drinking water treatment. Thank you.
All right, my name is Jen Title. I'm an assistant dean at the Graduate College. Thank you all for coming today. This was a fantastic competition. Before I announce the three winners, I'd like to give one last round of applause to everybody who was in this competition and did such a beautiful job. in our book, I mean, you're all winners here today, making it out of that original group. So it was really phenomenal and a pleasure to hear you all speak. So today we have one winner that will go on potentially to the MAGS competition, the Midwestern Graduate Association competition, to represent Iowa. We also have 2 honorable mentions. And I have forgotten the amounts on the awards. So our winner is $500, and our 2 honorable mentions will receive $250 each. Thank you. So our winner is Kather Ahmed. Would you like to stand up? Come on down. And our honorable mentions are Shi Yi Wang and Kathryn Peter.